The foundation of Dell platform is DNA-encoded libraries. Therefore, library quality is key. A high-quality DNA-encoded library means that the library compounds are in desirable property space with high chemical and topological diversity. That's because they support discovery programs of different modalities, help to generate reliable data streams for machine learning, pharmacophore paneling, and ultimately provide a rich source to elucidate structure-function relationship.
Hello, and welcome to Xtalks Spotlight, illuminating insights from subject matter experts and industry thought leaders. I'm Sonia Hunt. In this episode, we're exploring a critical question. What matters more in DNA-encoded libraries? Size, quality, or chemical diversity?
DNA-encoded library screening, often called Dell screening, is now a mainstay in early drug discovery, allowing researchers to test billions of compounds in a single experiment. As the technology has scaled, library size has often become the headline metric, with bigger libraries, broader searches, and more potential hits. But in practice, size alone has turned out to be a poor predictor of success. Very large libraries can introduce redundancy, background noise, and unclear signals. This can make results harder to interpret and more difficult to translate into confident medicinal chemistry decisions. In this Xtalks Spotlight, Ying Zhang, Vice President of Discovery at X-Chem, looks at why library quality and meaningful chemical diversity matter more than numbers alone. For discovery teams, this shifts the focus from how many compounds are screened to how well a library is built, helping reduce downstream risk and improve the quality of early starting points. Thank you for taking the time for the Spotlight interview, Ying.
Oh, thanks for your kind introduction, and thanks for having me.
So to start, Dell is often discussed in terms of scale, billions of compounds and massive numbers. Why do you think size became such a dominant metric? Is it perhaps a misconception? And what does that focus miss when applied to early discovery programs?
Yeah, it is true that DNA-encoded libraries can be numerically large. That's because the libraries are prepared in a split and pull fashion, in which the building block sets are introduced in a sequential reaction encoding cycles. So higher number of cycles effectively increase the numerical size of the library. However, it can negatively impact the physical chemical property profile of the library. So one simple example is when you add cycles of chemistry, it tends to increase the molecular weight of the compounds you produce in the library. In addition, adding cycles, or sometimes we call it steppy schemes, may also reduce the synthetic efficiency and potentially increase the side products that we're not intend to encode for. So what we want is library compounds in lead-like, drug-like chemical space. And that's why at X-Chem, instead of numerical size, we have been focusing on making DNA-encoded libraries with fewer cycles of chemistry while maintaining chemical diversity. So that's because we firmly believe that the library quality, including diversities and property profiles, are the most important factors in accelerating drug discovery. So actually, since 2017, other than macrocycle Dells, which we intentionally added to expand to beyond rule of 5 chemical space, almost all of our newly designed and synthesized libraries only contain 2 to 3 cycles of chemistry with an average molecular weight around 350 to 450. So this approach actually really pans out in our partnering discovery programs. We analyzed a large number, I think over 700 or so licensed compound families, and all clinical candidates are from those libraries with fewer than a billion molecules, which equates to 2 to 3 cycles of chemistry.
Now, when discovery teams hear the word diversity, they often think it just means different compounds. From your perspective, what does chemical diversity really mean, and why does it matter for partners running discovery programs?
That's a great question. So when we consider library diversity, we definitely think beyond their numerical size. So as you said, we don't consider this as simply another or different compound. We think the true chemical diversity comes from the library schemes, vector, geometry, like the 3D shape of the molecules, their connection chemistry that links the building blocks at each cycle of chemistry, and even the chemical diversity of building blocks themselves. So as a matter of fact, it is actually really important to take these into consideration at the library design stage. We do not want to limit ourselves to what can be made, but rather our library designs are guided by what should be made in order to deliver a successful discovery outcome. So, for example, we pioneered atom-efficient library design concept for DNA-encoded libraries and successfully adapt several small, compact aromatic and emphatic ring formation reactions on DNA, utilizing widely available building block classes to synthesize Dell compounds with high FSP3 characters and unique topological diversity. So compounds from these well-designed libraries display the most favorable physical-chemical properties to support small molecule drug discovery. So the goal here is what we want to achieve is when hits are identified from the platform, they are progressible in downstream hit to lead and lead to candidate campaigns.
Building on that, why is library quality so important for a discovery platform like Dell? And how does it affect the data you generate and the outcomes you see downstream?
The foundation of Dell platform is DNA-encoded libraries. Therefore, library quality is key. So quality goes beyond library purity, chemistry and encoding fidelity. A high-quality DNA-encoded library means that the library compounds are in desirable property space with high chemical and topological diversity. That's because they support discovery programs of different modalities, help to generate reliable data streams for machine learning, pharmacophore paneling, and ultimately provide a rich source to elucidate structure function relationship. So Dell platform is a perfect union of chemistry and biology, and it really generates data at an omics scale. So we call this chemomics. So data quality such as consistency and data diversity is particularly important for machine learning, and this can only be achieved from a high quality source. So we actually reported in a seminal 2020 Dell ML paper in collaboration with Google, where we evaluated and concluded that Dell data generated from our DNA-encoded libraries is a reliable source of machine learning data. And this was further illustrated in the subsequent collaborative work with SGC. We reported a high success rate of hit identification against an array of, I think, 18 targets of the WD40 protein target class, utilizing machine learning from Dell data alone and generating first-in-class and highly diverse tool compounds and hits for a very difficult target class. So all this speaks to the quality of Dells being a key to unlock successful outcomes. And it isn't a numbers game. the quality of the inputs really determines the quality of the outputs.
Taking this a step further, how does library design influence early discovery decisions? And how does it affect overall program risk?
I just can't emphasize enough that the library quality, such as compound diversity, really impacts the outcome of discovery. So over the years, we have received lots of feedback from our discovery partners that our Dell hits bind to cryptic pockets, or they identify allosteric binders, or inducing unprecedented conformation changes of the protein. So we hypothesize that chemical diversity must have matched biological diversity. So therefore, a truly diverse library, chemical library, should be able to engage more targets and do so in novel ways. So this isn't just our experience. We also surveyed the Dell hit ID literature, like hit identification literature, and found this to be the case for other well-designed Dell campaigns as well. So this observation actually led to a perspective article in 2024 featuring industry opinion leaders. The article proposed our view of library quality and chemical diversity from a structure biology point of view, and how these factors impact the discovery outcomes as measured by head identification for desired modes of action. So screening a highly diverse Dell collection really helps us to deliver programs across various target classes. So in our case, including very difficult target classes such as protein-protein interaction, modulators of induced proximity, receptors, with a well-designed Dell campaign, even for DNA binding proteins as well.
And for teams evaluating Dell partners, what questions should they be asking about quality and diversity beyond simply how big is your library?
I think you have asked many great questions already. These are exactly the questions I would have used to evaluate Dell partners. So beyond size, for example, you should ask how diversity is defined, how it is measured. This should give a glimpse of whether the library is designed with the downstream work in mind. Another useful question is perhaps how often your results lead to follow-up programs. Perhaps look at what kind of compounds they publish, which indicates the novelty of library schemes, library design consideration, physical chemical properties of the library compounds. So we actually analyze our reported compounds in X-Chem's publication since 2020 and calculate the average physical chemical properties of the compounds we publish. So the average molecular weight is just above 400, with an average number of amide bonds below 1, which really means the library compounds identified from our libraries are actually in a highly desirable property space. And this, I think this ultimately increases the progressibility of the Dell hit compounds in the discovery pipeline. Our collaboration partners are able to develop these hit compounds to clinical candidates, with the most advanced in phase 3. So I think these questions really focus on whether the data supports confident decision-making, and it all boils down to how they support the desired outcome of your discovery programs.
And finally, what's the key takeaway you want our audience to remember from this discussion?
I think the quality of DNA-encoded libraries such as chemical diversity and the compound properties really greatly impacts the outcome of small molecule drug discovery. So X-Chem, as the leader in DNA-Encoded library design is happy to be partnering with you to achieve your goal in your early discovery programs.
Well, thank you very much, Ying, for speaking with us today. We really appreciate your time and insights.
It's great that we chat about Dell, and thank you again for having me.
We look forward to seeing how a continued focus on Dell library design supports clearer, early decisions across drug discovery programs. Thank you all for joining us for this X-Talks Spotlight. We hope you enjoyed the discussion.